All right, so hopefully people took a look at the heart anatomy and some of the cardiac cycle. Even if it didn't make sense to you, hopefully we'll be able to make a little bit more sense of this. Today we're going to spend just a brief moment looking at heart anatomy, but we're going to do more of a focus on the heart anatomy from the cardiac cycle perspective. All right, and so we're going to spend a little bit more time with looking at it like that. So remember here, the heart is really the main pump of the cardiovascular system, and it's pumping blood really to two major areas. All right, so one place that it's pumping blood to is the pulmonary circuit, right? And so that's the lungs. The other major place is to the rest of the tissues, the systemic circuit. Those are the two main places that the heart is. Blood is going to be coming in into the right side of the heart and then pumped to the pulmonary circuit. And then in the pulmonary circuit, that less oxygenated blood is going to become more oxygenated, right? So that's what we have with gas exchange that occurs in the lungs. That more oxygenated blood is going to come back to the left side of the heart, and then the left side of the heart is going to pump it to the rest of the systemic tissues, the systemic circuit. Okay, so even though we have two sides of the heart, the right and the left, okay, the blood doesn't, under normal circumstances where someone doesn't have some sort of defect, the blood doesn't transfer the other side of the heart. Okay, right? So one side of the heart's actually pushing blood to one circuit, the other side of the heart's pushing blood to the other circuit, right? So one of the things you want to be able to do is, okay, well, if you have a red blood cell and pick a place, so if it starts here, okay, try to trace the path that red blood cell is going to go. So it's going to go through and be able to name these of the anatomical structures that it's going to pass through. All right, so that's one of the things that you're going to want to be able to do, right? So be able to track or trace, the path of red blood cell goes if you pick a place and then follow it through the circuit. Okay. All right. Okay, so this is the other anatomy that we're going to talk a little bit about. So hopefully people remember from Bio 241 when you guys talked about membranes, right? So here we have a serous membrane, and one of the things about a serous membrane is we have that membrane kind of folds back on itself, right? Okay. So when we talk about a serous membrane, it's like you are pushing something into like a balloon, right? And that membrane, right, part of that membrane is going to be closely associated with whatever the organ, all right, that it attaches to. That's called the visceral membrane. And then the outer portion is called the parietal, right? So if we're taking a look at the pericardium here, all right, so we have a visceral pericardium. So the visceral pericardium here, which is tightly associated with the heart, and it circles back around, all right? And this is the parietal part of the pericardium that's lining the cavity, okay? And then we have some fluid in between this pericardial cavity that allows these membranes to slip past each other. And that's really important because these organs, they actually move. In the case of the heart, it's beating, right? And so here we have a specialized term for this visceral pericardium. It's called the epicardium. Okay. Underneath that, we have the myocardium. And so that's the cardiomyocytes. That's the cardiac muscle tissue. All right. So every time you see the word myo, it's referring to muscle. All right. Now we have a membrane that's on the inside that's lining the chambers of the heart, and that's the endocardium. Right. Okay. And so when you guys do the heart dissection, you guys are going to be seeing the parietal and the visceral pericardium. The visceral pericardium is going to be tightly associated with the heart. Okay, so the epicardium there. So keep that in mind. Most of the tissue that's making up the heart is going to be cardiomyocytes, okay? Cardiac muscle tissue. All right, so some of the major structures here. So for the heart chambers, we've got 4. All right, and remember here, we always refer left and right are from the subject or patient's perspective. So we have the left atrium, left ventricle, right atrium, right ventricle. If you are taking a look at the heart from a superficial view, okay, this is going to be the right side of the heart. This is going to be a left side of the heart. Up here is where we have the right atrium. Over here is going to be where we have the left atrium. And then down here is where we're going to have the right ventricle. Then over here is where we'll have the left ventricle. Okay? It'll be easier to see once we take a look at a section of the heart, when you look inside. Okay? So major blood vessels. So major blood vessels are going to be taking blood to the heart or away from the heart. So here, veins are going to be taking blood to the heart. Arteries are going to be taking blood away from the heart. And so here, we're going to have the vena cava, superior and inferior. Those are some of the major venous blood vessels that are taking blood to the heart. And then we have aorta, which is the major blood vessel taking blood away from the heart. Okay. One of the other things that we have here is something called auricles. These are kind of ear-like protrusions that increase the size of the atria. Okay, so remember the right atria was over here, and these are kind of ear-like protrusions that kind of increase the size of the atria, so they have a slightly larger volume. Now, as I strongly suggest, when you guys look at a model, you want to take a look at it from multiple viewpoints, right? So this is an anterior view. Here we have a posterior view that's going to help with looking at seeing some of these other structures more easily, right? So here you can see the left atrium a lot easier, and you can see some of these other arteries and veins, okay? So here we have pulmonary artery, and then we also have pulmonary veins. So the word pulmonary reminds you of what organ? Lungs, right? And if we're talking about a pulmonary artery, is that blood going to or from the heart? From the heart. It's going away from the heart. And if it's a pulmonary artery, where's it going to? The lungs, right? So pulmonary vein would be, yeah, so basically going from the lungs to the heart, right? So here, hopefully now people can take a look back at this and start realizing that, oh, yeah, that makes sense. This is a pulmonary artery. And then here I have pulmonary veins, right? So if you take a look at this, the pulmonary veins, or do veins in general, always have less oxygenated blood than arteries. No, if you look at the pulmonary circuit, they're switched, right? So a lot of times, you know, students think that just because it's an artery, it always carries the higher oxygenated blood. That's not true, right? So if you take a look at the pulmonary circuit, you know, the pulmonary artery is actually carrying the less oxygenated blood than, say, the pulmonary veins, okay? The bottom line, though, is arteries and veins. What do they always do? Arteries are taking blood away from the heart. Veins are taking blood to the heart. All right, so like I said, we'll get to see some of these chambers a little bit easier once we have a section through them. And so once you do that, you can see that we have, this is the right atrium, the right ventricle, left atrium, left ventricle, okay? Now, one of the things is the heart pumps blood in a direction, right? Pumps are, this pump that we have is pumping in a unidirection, in a single direction. In order for that to happen, we have to have valves that prevent blood from going back in the wrong direction. And so here we have valves that separate the atria from the ventricles, Okay? And here, this is one valve that separates the right atrium from the right ventricle. Here's another valve that separates the left atrium from the left ventricle. Since the valves are separating atria from ventricles, they're called atrioventricular valves. All right? We have one on the right, one on the left. The one on the right is going to be called the tricuspid valve. The one on the left is called the mitral valve. All right? It's the mitral valve is named because it resembled the shape of the mitre, which is the hat the Pope wears, okay, with the two leaflets. The way I always remember it is mitral has an L in it, and that's on the left. All right. So we have two valves here, atrioventricular valves separating the atria and ventricles. You can see also here that we have these cords that are attached to the valves. And these cords are called chordae tendineae, and they're attached to these muscles. Okay? As we'll find out that these ventricles, when they are going to be propelling blood from the heart, they're going to squeeze, and they generate a lot of pressure on these valves. So these are kind of like parachute cords to help stabilize the valve so that it doesn't prolapse back into the atria. Okay? Now, we have another set of valves that are called semilunar valves. So there's one here that separates the ventricle from the blood vessel taking blood away from the heart. There's another one over here that we can't see very well on this image or this view that separates the left ventricle from the aorta. They're called semilunar valves because they have a half moon shape to them. So they're cupped like this. And so we'll find out that that's really important in just a second, because if blood flows back here, it is going to gather in these cusps, and cause these valves to close. All right. So here we have two atrioventricular valves. We also have two semilunar valves. All right. The two semilunar valves we have, one is called the pulmonary valve. The other one's called the aortic valve, named for the blood vessels that the blood's going to go into. Let's see. Separating the two ventricles is the septum here. All right, that's the interventricular septum. And then there are the trabeculae carneae, okay, the ridges here in the ventricles. And that's it's thought to help with, if you squeeze the ventricles together, the walls don't stick together. So think about if they were smooth, all right, and you have something liquid between them. Think about if you have two glass pieces and you put a little bit of water in between those smooth glass pieces. Really difficult to pull them apart, right? But if you put like a little bit of sand or something, to help separate them so it's not smooth, it's much easier to separate those two glass pieces, if you have a little bit of water. And so that's what's thought to be the function there of the trabeculae carneae. All right, so let's quickly go through here. So you want to keep in mind, where is blood going and for what purpose? Some of the major blood vessels that we just talked about. All right, the valves involved in the cardiac cycle, and the four chambers of the heart. Hopefully those are already in your mind from what we just talked about, right? What is happening during the heart sounds and thought to contribute to the forming of those heart sounds, okay? So here, the cardiac cycle is the physical movement of blood through the heart, all right? It's the physical movement of blood through the heart. It's the mechanical contractions that we're talking about, right? And so here, just remember, arteries are carrying blood away from the heart, veins are carrying blood to the heart chambers. And we've talked about this already. And so here, the superior inferior vena cava, and then we have the right atrium, right ventricle, left atrium, left ventricle. Then we have the tricuspid valve, one of those AV valves separating the right atrium from the right ventricle. And then we have the mitral or bicuspid valve that's separating the left atrium from the left ventricle. And then we have the chordae tendineae, which are those parachute cords helping stabilize the AV valves when there's a increase in pressure there from the ventricles contracting. All right, and then there's some valves that we can't see very well. The pulmonary valve we can see, and the aortic valve we can't see very well. And this is just saying that the pulmonary trunk is part of that pulmonary artery taking blood away from the heart into the lungs. Okay, and there's our aorta. Okay, so some of the major blood vessels and valves involved. So, like I said, the aortic valve. Why is that? Well, it's because the aortic valve sits behind the pulmonary valve. Okay, so you couldn't see this guy because it's kind of obscured in that frontal view, by the pulmonary valve. And remember, both of these guys are semilunar valves, and those mitral and the tricuspid valves are AV valves. Okay. All right. So cardiac cycle, squeezing or contraction. That's called systole. So S, systole, squeezing, S. All right. Diastole is relaxation. Whole principle behind movement of blood through this is that blood, substances such as blood flow down their pressure gradient. They go from high pressure to low pressure. All right. From high pressure to low pressure. Okay, so what ends up happening is that we start getting the atria filled. All right, blood coming into the atria, and it's going to flow down its pressure gradient. It's going to go from high pressure to low pressure, and it does so passively. Okay, so here there's passive filling of the atria that goes on. And since those AV valves are open, a lot of that blood is going to go from the atria and start filling the ventricles. Okay? So at this point, a lot of times when we talk about systole and diastole, we're really interested in the ventricles. All right? But we can also talk about systole and diastole for the atria as well. Okay? So here, if we want blood to flow down the pressure gradient, we have to make sure that the ventricles are going to be relaxed. If they're not relaxed, then the pressure in the ventricles is higher than in the atria, and blood's not going to flow down its pressure gradient in that case. So we start off with ventricular diastole, all right, such that we're going to get filling of the ventricles. And what's going to happen is the ventricles are now going to start squeezing. They're going to undergo systole. Okay? So when they start squeezing or contracting, what's that going to do to the pressure in the ventricles? It goes up, right? So here, now we have increased pressure in the ventricles such that it's higher in the ventricles than it is in the atria. Right? Which means blood's going to push up against the AV valves, causing them to close. Right? Now at this point, the pressure's not high enough, okay, in the ventricles compared to the other side of the pulmonary arteries. So these valves don't open yet. So here we have the AV valves closed and the semilunar valves closed, right? Which means that there's no place for the blood that's in the ventricles to go yet, right? But we're still increasing the pressure in the ventricles, okay? Does that make sense to folks? So we're increasing the pressure, but we haven't increased it to a point where a valve is opened yet. We've only increased the pressure such that we've closed the AV valves, okay? So here, because the volume doesn't change, sometimes this is called isovolumetric. Right? Iso meaning the same volume. So we call this isovolumetric systole, ventricular systole. Okay? Does that make sense to folks? So it's iso because the volumes are the same. All the valves are closed. The blood doesn't have any place to go yet. Okay? but the pressure is increasing. Eventually, the pressure, the ventricles squeeze, get to a point where the pressure in the ventricles is much higher than on the other side of these semilunar valves, and that's going to cause these semilunar valves to open, allowing blood to escape from the ventricles. Right? So here we go from ventricular systole, which is really an isovolumetric contraction going on, where we have all valves closed, but the pressure continues to go up, and eventually it goes to a point where that causes that semilunar valves to open by pushing blood through them. Okay? All right? So here we have ventricular systole going on. In this case, we're actually ejecting blood from the heart now. Now, what ends up happening next after blood gets ejected from the heart, okay, those ventricles are in systole, but then they go into diastole, start relaxing. But we just pushed a whole bunch of blood, say, into the aorta and the pulmonary artery here, okay? Which means that if these ventricles start relaxing, and we just have a bunch of blood just on the other side of these semilunar valves, okay, where do we have our pressure gradient now? So it's going back, right? We have more pressure on the other side of the semilunar valves than in the ventricles. Does that make sense to folks? Which means that's going to allow blood to flow a little bit back in the other direction, and it's going to fill those cusps of the semilunar valves. And when that blood fills the cusps, it causes them to close. All right? Does that make sense to folks? So this is all dependent upon pressure. And how is pressure changed? Well, pressure is changed by whether you have systole or diastole going on, right? So here, initially, during ventricular diastole, okay, the atria are going to allow blood to fill into the ventricles passively. And at the very last bit, the atria contract. So we have atrial systole, okay, that goes on. And then the atria relax. Then we have the ventricles contracting. And then we have the atria contracting, and then the ventricles contracting, okay? All right. So let me see if this works. So notice that both sides of the heart, ventricular systole and diastole, happen at the exact same time, right? So it's not that we get the right and then the left, the right and then the left. No. It happens at the exact same time. So we get ventricular, so we get loading of the atria, okay? And then we have the blood flowing from the atria into the ventricles, and then from the ventricles, it's going to be pushed out via the exiting blood vessels, right? And I'll get to your question in just a second, all right? Does that make sense to folks? All right? Okay, so one of the last things, or one of the other things, is that when the heart contracts, it actually twists a little bit, all right? So if you want to take a look at some of the open heart surgery videos or of a beating heart, you know, online, you will see it twist. And the reason for that is because of this, what's called the myocardial vortex. As we go from, up here is called the base, down here is called the apex. As we go from base to apex, the myocardium twists like this. And it turns out the chambers also twist. So if we take a look at the ventricles, they actually turn like this as you go down towards the apex of the heart. So it's not like the ventricles stay like this. They actually twist around each other. And when you guys do a heart dissection, you'll kind of see this. All right? Okay. Sorry, go ahead, Marshall. Yeah, so basically the whole idea is when the ventricles are in diastole, all right, you can have the atria in systole. It's a bad thing if you have both of them in systole, you know. That's right. Right. Most of the blood that goes from the atria into the ventricles, it fills the ventricles passively, and then the last little bit is actually pushing the ventricles using atrial systole. Okay? Yeah. Yeah, go ahead.
What would happen if one of those AV valves don't close? Like I'm supposed to...
It's a good question. Yeah, so if the AV valves don't close or if they have a prolapse or something like that, you know, I think we're going to have a good discussion on Thursday about that, right? Yeah. Now, the other question is, where do we get the heart sounds? Turns out the heart sounds happen around the time the valves close. All right? So take a look at when the valves close. First, which set of valves close? The AV valves, right? So that's the first one. And it's really powerful, right? So the first heart sound, S1 in this case, that's a really much more prominent sound when you listen to the heart sounds. What about the second heart sound? Well, that's when the semilunar, or around the time the semilunar valves close. Okay, and a lot of people, you know, attribute the sounds to the actual closing of the valves. You know, there's some studies out there where they put microphones inside the blood vessels and they've timed it. And it turns out the sound seems to come just slightly before the valves actually close. So it's thought to actually be the blood rushing up against the valves, pushing out against, you know, some of the walls of, say, the blood vessel or the heart wall. Or, you know, for the first heart sound, it's the tensing of the chordae tendineae. So if you pull a string really quickly, it makes a sound. You know, so all of those things together are thought to contribute to these heart sounds that we hear, and also the heart pushing against the pericardial cavity. All right, so we'll take a break after this slide. Just putting some of these things together, right? So here we have blood that's going to be filling into the ventricles via the atria, right? Both in the right and the left. And so here, ventricular filling, and so we have this as rapid filling, and then we have atrial, the ventricles are in diastasis, right? So they're relaxed. And then you have also atrial systole to push a little bit more blood into the ventricles, okay? Right, so you can have atrial systole at this point to push a little bit more blood into the ventricles. And then we have ventricular systole that goes on, and the pressure increases in the ventricles to a point where you get these AV valves to close, but not to a point where the semilunar valves open. Right? So if all the valves are closed, right, we have isovolumetric, right? It's the same volume. That blood that's in the ventricles isn't going anywhere. But what ends up happening is the ventricles continue to squeeze, increasing the pressure in the ventricles, and eventually it gets to a point where those semilunar valves are pushed open. Right? And then blood gets ejected from the ventricles, right? And then we have isovolumetric relaxation that goes on where we basically are going to get blood filling the atria, and eventually that pressure in the atria is going to be greater than the blood or the pressure in the ventricles, causing the AV valves to open again, and we start this whole process over, right? And so here, if you're taking a look at this, where do we have our heart sounds? So S1 is going to be when these valves close. Isovolumetric contraction is starts at that point. And then here, when we have ventricular ejection going on, we're going to get these valves to open. And then when we have the S2 is going to be when we have this isovolumetric relaxation going on, right? Okay, so basically, you know, the valves opening and closing is right around the time when we have heart sounds. Okay, all right. So we'll take a 5-minute break, and then we'll continue taking a look at the heart after that. All right. So I just wanted to play. Hopefully the audio works. You know what a normal heart sounds like. So you guys can hear the lubbed up. So the first heart sound, that's around the time that the AV valves are closing, and the second one is around the time that the semilunar valves are closing, right? Okay. So for whatever reason, the sound's not working there in the lab, so we have to listen to it here. And I think, so this is a 1-year-old. Much faster, huh? All right. So one of the things that I'm actually happy to see on this cartoon, it's actually kind of nice, is that after ventricular systole, so after the squeezing of the ventricles to eject blood from the ventricles, right? You see when the ventricles are relaxing, have they ejected all of the blood there? No, some is left over. Okay, so it turns out the ventricles, when they eject blood, don't eject all of it. Some of it gets left there. Okay, now how can we figure out how much blood was ejected, right, from the ventricles? Well, the way to do that, okay, is to say, well, how much blood did I start with in the ventricles, right? So how much blood did I start with in the ventricles? Well, the total amount of blood I had in the ventricles to begin with before ejecting blood, that was at the end of what? Yeah, end of diastole for the ventricles, right? So that's called end diastolic volume. Right, so end diastolic volume. So that's the total amount of blood I have in the ventricles, right? Okay. Well, if I want to know how much blood was ejected from the ventricles, I could take my total amount of blood I had in the ventricles and then subtract the little bit of blood that's remaining in the ventricles after systole, right? So if I start off with something that has this much in it, okay, and then I'm left with this much, if I take the difference between those two, I know how much I took out. Does that make sense to folks? That's exactly what we're doing here, right? We're saying we started with this amount, I got rid of some of it, and this is what I'm left with, okay? How much did I get rid of? I can subtract the two, right? So here I have end diastolic volume. So after the end of systole, okay, I have a little bit left in the ventricles, right? A little bit of blood volume left in the ventricles. That's end systolic volume, right? The difference between the two is how much blood got ejected from the ventricles, or what we call stroke volume, right? So that's how much blood left the ventricles when the ventricles went through systole. Does that make sense to folks? Why this equation works? Okay, we end diastolic volumes, total amount of blood that I have in the ventricles. Okay, and systolic volumes, a little bit of blood that I have left over after we've ejected blood. Right? That's the stroke volume.